Alright, so you should have picked up in class, or if you weren't in class, then you can get the worksheet on Blackboard. We're going to take a look at Monohybrid 2 worksheet, and these are going to be examples where, for the most part, we work backwards, meaning that we have the genotypes of the kids, and we're trying to find what the genotypes of the parents are, or we have the phenotypes of the kids. Either way, some key things that we need for monohybrid practice, or monohybrid problems, or really problems in general, is that we need to remember that parents are going to have 2 alleles per trait, really everyone. So everyone will have 2 alleles per trait. So this means if you have brown hair, you have 2 alleles that gave you that brown hair. Now, whenever we are talking about genetics, we talk about how parents are going to just give 1 allele to each offspring. So they give 1 allele, or 1 letter as we've looked at it, to the offspring. Then, just from that, we can say that offspring will have 1 allele from each parent for 2 total alleles. This will just be helpful to us whenever we are looking at these problems so that we can remember how we're going to be working backwards. Alright, so we're going to go ahead and take a look at problem number 1 on the Monohybrid 2 problems. We're going to follow kind of the same way where we've written our knowns and our unknowns and work in that direction. So whenever we're looking at number 1, in problems 1 and 2, it says assume that in some insects, long-winged is dominant to short-wing. So we're going to say known, and we're going to say that homozygous dominant or heterozygous is long-winged, and then homozygous recessive is short. Problem number 1 specifically says if 50% are heterozygous long-winged, so we know 50% are hetero-long, we have 25% that are homozygous long, and then we have 25% that are short. From that, we can go ahead and assign the genotypes of each of those offspring. And so this is our offspring here. And so we can assign our genotypes. So heterozygous would be capital L lower, homozygous long would be capital capital, and then short would be lower lower. Our unknown and what we're trying to find are the genotypes for our parents. So our unknown are the parents. And I set them up like this. So 2 lines because they each have 2 alleles and they're crossing with another parent that has 2. So each offspring is going to get 1 allele from each parent. So whenever we have homozygous long, that means, so we can look at heterozygous and we can say that they have a capital L and lower, so each parent is contributing something, but we don't really know where to put that. So it's best to start with the homozygous characteristics. So if we have an organism that has 2 capital L's, that means that each parent had to have had a capital L for them to have 2 of them. Whenever we look then at the homozygous recessive, or really anytime we have a recessive trait, we know that each parent had to contribute a recessive allele to their offspring. So this really would be our answer. I'm going to show you with the Punnett square and just prove that this works. And so we'll do the Punnett square of the parent. And you can do this too, just to support your answer. So if we look at this, and we're looking at our genotype ratios, we're going to say that we would have 1 homozygous dominant for every 2 heterozygous for every 1 homozygous recessive. So that would be 25%, 50%, and 25%, which matches up with what we're given. All right, for the next one, which is number 2, we're using the same information, so we're saying that capital L, or capital lowercase is going to be long, and then lowercase is going to be short. In this case, we're saying that 50% of the generation, or the offspring, are short-winged. And we know that that means that their alleles are going to be lowercase l, lowercase l. That also means that 50% of the generation is long. And so that means that their alleles would have to be capital L, and then it could be either capital or lowercase, we don't know. So our unknowns, so I'm going to just note again that these are the offspring. Our unknown are the parents, which again, they have two sets of alleles. So from here, we know that we have little l, so each parent has given a little l. One of the parents at least has to have an uppercase L or a dominant allele in order for this to work. So the question is this blank space, so is the blank space that's right here, is this going to be capital L or, it'd be helpful to get this, capital L or lowercase l? So you can kind of guess and check based on what we did before, or you can do two Punnett squares to show this. I'm going to go back to our previous problem where we did a heterozygous cross, and just note that this gives us 25%, 50%, and 25%. So here I'm going to go ahead and assume that this is going to be the recessive allele, and I'll do the Punnett square just to prove that it is. So whenever I do the Punnett square, I should find that I have 50% of my offspring are going to be short, or rather, sorry, these are short wings, so 50% of the offspring are short-winged. So this matches what I was given, and so I know that it's the correct answer. All right, lastly, I'm just going to talk about number 6. So number 6 is asking you to do a few Punnett squares, and it's using tall and dwarf plants. So in part A, it's asking you to find the F1 offspring. This is going to be a result of the cross of two parents. So you could think of this as the grandparents, and like your grandparents cross, and then you have the F1 generation, which would be your parents. In part B, it's asking for the F2 generation, and so that's going to be your F1s times your F1s, and that would be your F2 generation, so you can think of parents crossing, and then you are the F2 generation. All right, for part C, what we're looking at is we're looking at the F1 progeny, and so we are crossing, or F1 offspring, so we are crossing the F1 with the tall parent, and then in D, we're crossing F1 with the short or the dwarf parent. Part E is looking at a test cross. This is how a scientist can test an unknown dominant phenotype. So this is how to test an unknown dominant phenotype. So you're going to do two Punnett squares. So if the dominant phenotype was homozygous dominant, then you'll do a Punnett square to show that and report what the phenotypes are. And then you'll do heterozygous with a recessive. And so whenever this happens, scientists use a recessive and they cross it with an unknown dominant. For all of these, you're going to report the phenotypes of the offspring. So you want to report the phenotype ratios or the phenotypes of the offspring. Okay? Again, a test cross is with a recessive, crossed with a recessive. So give these problems a try. If you have any questions, please let me know, and we will go over them again in class.